Hello, everyone, and welcome to this library webinar. My name is Mads. I'm from the Reshape marketing team, and I sit here in the same room, in this very small room with MA.
Hi, everyone. My name is MA. I'm product lead at Reshape.
And MA, if there's any product-related questions that you want to drop in the chat during this webinar, MA will be here to assist answering those. But other than that, we'll hand over to Jen, my colleague, who's going to be running the webinar. So Jen, take it away. And just for everyone, drop your questions in the chat and we'll record this session and send it to you afterwards. So if you miss parts of it or have colleagues who want to see it, you'll have it shortly after this. All right, Jen, take it away.
Perfect. Thanks, Mads.
And there we have it, the presentation.
Can you hear me?
Yes.
Yeah, I'm getting a bit of reverb. Okay, real good. And can you see the slides?
Yes.
Perfect. Okay, so yeah, thanks to everyone joining today for taking 30 minutes to hear a bit about our updates here at Reshape. I'm probably a new face for some of you, so I thought before we jump into the webinar, it might be good to briefly introduce myself. Now I'm part of the marketing team, but I've been spending many years working in the lab across a range of different fields. I've worked from insect research, microbiology, and vector-borne disease research. I've also worked in labs across 3 different countries. So I'm originally from South Africa, but I completed my PhD in France and then did a postdoc here in Denmark. And one thing that I've learned along the way is that lab life is its own international language. No matter where you are, we all face many of the same challenges, whether that be trying to make collaboration easier, keeping better track of our experiments, sharing results more smoothly, and honestly, just trying to fit biology into something close to a 9-to-5. And that's actually why I was so happy to be introduced to Reshape a little over 4 years ago now as a tool that I was using in the lab. And now it's great to be part of the Reshape family, and it's incredibly exciting to help show the future of what we're creating for scientists. Okay, so just jumping right into it. What you can expect for the next half an hour is around a 20-minute overview of why we do what we do, touching on the power of fully integrating biology with AI, also the importance of maintaining structured libraries of your data. We will also have a brief showcase of the end-to-end workflow, as well as a case study highlighting an example of where we're going next. Finally, ending off with the bigger picture for why we're doing all these updates. Okay, so the year is 2026, and we're surrounded by a huge wave of companies using AI to build tools that streamline the way we work, and biology is no exception. The generative AI in biology market is expected to reach $356 billion by 2032, and we're already sitting on a $142 billion market. We've already seen major breakthroughs making headlines, and a great example of this is AlphaFold, which used AI to predict and map over 200 million protein structures in only 4 years. Before that, around 170,000 protein structures were built up slowly through experimental work, one at a time, from data that was scattered across public databases. The point here is that in a remarkably short period of time, AI transformed what used to be a slow sequential process into something that can now be done at massive scale. So we now have AI helping to drive breakthrough discoveries in biology, but the big question for us at Reshape is how do we actually bring that power into the wet lab? We're seeing deeper insights become more and more accessible through AI, but there's one requirement that never changes, and that is that AI is only as good as the data we feed it. Because at the end of the day, data is everything, right? And without a solid data foundation, it's the classic case of the garbage we put in is the garbage we get out. And a lot of labs are already recognizing this, which is why we're seeing a growing adoption of lab management tools, and software designed to consolidate, organize, and share experimental data more effectively. Chances are your lab is probably already looking into this and in the process of adopting a lab management system. And that's definitely a step in the right direction. It helps structure data once it exists, but it still doesn't solve the whole problem. And here I would argue that the real bottleneck is actually how we are generating data in the wet lab in the first place. And until we improve that process, we're limiting the quality and consistency of our data, and then ultimately limiting what AI can truly unlock for us. As I mentioned, we are often trying to fit experiments into a 9 to 5, but in reality, biology doesn't work on our schedule. It happens around the clock, and while we're only checking experiments every 12 or 24 hours, we may be missing biologically meaningful insights happening in between. So what we really need is a full end-to-end workflow, one that automates data acquisition, captures the right metadata as we go, and generates immediate insights in real time. And that's how we build the kind of quality data foundation that makes AI genuinely powerful in the wet lab. And just to briefly build on that point, think about the assays you are currently running in the lab, and then think about how you actually share that data with your collaborators. So often, we're still relying on what we like to call artisanal data generation. These are things like manually counting colonies, recording growth rates by hand, or visually scoring assays. And if we're lucky, that data eventually makes it into a shared drive, but often without the extra context that makes that data truly meaningful. And that's also before we factor in variability across time, across different people, and across different ways of doing the same type of experiment. And in my opinion, this kind of fragmented and poorly maintained data is expensive. It's expensive in time, in money, and more importantly, it's costly in terms of the insights we could be extracting but simply aren't. So when streamlined data collection is paired with rich and consistent metadata, we unlock faster learning cycles and accelerate hypothesis testing. But that, of course, requires moving beyond siloed assay data and consolidating data across sites, teams, and time. Many organizations are already sitting on gold mines of data that they just can't properly utilize. So what's needed instead is a connected data foundation, so a unified searchable data lake where results and metadata can be compared and turned into insights at scale. So what is Reshape's solution to solve this problem? If you don't already know Reshape, we've gotten really good at capturing plate-based assays and using AI to extract highly accurate and standardized measurements. Now with the Reshape library, we are building on that captured data to easily aggregate insights and navigate through all your dimensions of data, like conditions and treatments. Of course, that relies on accurate metadata, which is why we also recently released the Plate Designer feature to help you design and input your metadata to keep track of all your assays. Okay, so I think this is a good time to jump into how this end-to-end workflow looks in the Discovery platform. And maybe some of you are already familiar with the Discovery homepage, where you can see an overview of your devices, some simple metrics like what device is running and total images taken. But for now, I will jump straight into the Plates feature, as this is where you will input most of your metadata before actually running your job or assay. So the setup here is really simple. Now you're seeing an overview of all of my plates. But to set up a plate, you simply find and select Create Plates. Then you'll be redirected to a page where you can customize and design your plate. Reshape is compatible with all plate formats, so you can choose between any plate type. But for this example, I configured an entire tray of 6 well plates. So step 1 is choosing the plate type and quantity, and step 2 is to input the metadata or well properties. So this plate is from a job I was running for a preservative efficacy challenge test, which is similar to what one would run in a food R&D lab. For this job, I was looking at 2 spoilage organisms on yogurt. And so once the plates have been populated with the properties, so the well properties, we can just simply select create plates. Then it is time to actually run a job, which is just Reshape speak for running an assay. And here you can see an overview of my tray and plates. And what's really important at this point is that I associate each plate with a plate map referencing all of my metadata. And this can be done via the drop-down menu, or if your lab is equipped with a barcode scanner, you can simply scan each plate. You then input all the final parameters of your assay or job, including temperature, analysis type, capture rate, before finally starting your job. Your data is then captured automatically 24/7 without the need for any further manual monitoring. And once you run all your jobs, you can consolidate them all in a single project. So this is my project, consolidating jobs across all of my preservative efficacy challenge tests. As I mentioned, I looked at the growth of 2 spoilage fungi, so it was Penicillium and Fusarium, and this was on yogurt with a specific preservative concentration across a range of temperatures. In practice, this is just a kinetic growth assay that measures the time to appearance and growth dynamics of these indicator colonies. This allows us to evaluate the impact of temperature on preservative efficacy. So this job was run 7 times at different temperatures. And now what you're seeing is the library feature, which gives you classification information and along with your metadata maintains a record of the projects and jobs the organism has been used in. You're essentially building here a searchable data set of every organism your team has ever worked with, and the ability to generate deeper insights with every new experiment. Along with basic information like organisms classification, you can also start to aggregate insights across jobs. So if I go back to my project, I collected data in each well of all 10 plates across 7 jobs. That's a total of 420 wells that I'm using to aggregate insights. So this is the power of the end-to-end workflow. I can easily unlock insights across all this data I've collected. And what that aggregated data looks like for this type of experiment is that we can immediately extract insights such as the time of appearance of the inoculated challenge organism, in this case focusing on the Penicillium, across a range of different temperatures. And this is almost as soon as the assay has been run. We can start to see patterns and acquire instant insights that can be quite cumbersome to analyze when results are stored in isolation. For example, here we can see how small temperature changes the time to appearance, revealing temperature-sensitive preservative performance. Or if we included different preservative concentrations, we could see how spoilage organisms behaves across different yogurt formulations. So this is where we're at today, but we are rapidly moving forward with all these insights. Now, if you run challenge tests at a few key temperatures in combination with different challenge strains and preservative concentrations, you can start modeling what might happen at intermediate temperatures or under slightly different storage scenarios without needing to run every possible experiment from scratch. In practice, you'll be able to forecast outcomes under new or untested conditions. So this leads into predictive insights, which we are starting to run with some customers collecting the appropriate data. In this example, we ran an internal study focused on optimizing antifungal performance across key conditions. This was compound strength, pH, temperature, and various strains. Here we have an overview of all the parameters, as well as the organisms associated to the study. Below you will see an overview of the core insights. The green indicates that the conditions meets the shelf life target, while the red shows it would probably spoil before reaching that shelf life. Again, these are insights that are immediately aggregated and accessible once you've input your metadata and run your assay. Furthermore, based on your previous assays, the platform could suggest the best formulation for your required shelf life product or period, as well as give a list of viable formulations needed to reduce the microbial load according to the longevity and temperatures of your product. So all in all, this will allow teams to test and simulate a wide range of parameters digitally and upfront, which has the potential to dramatically narrow the research space to the most promising options, essentially adding a new but faster screening layer to the R&D funnel. And so zooming out of all of these specific examples, there is so much potential with the Reshape library, and it's continuously expanding with capabilities like this, directly estimating your final concentrations from plate counts. But the possibilities don't stop there. So for agri-labs, you could predict insecticidal activity, you could aggregate insights across strains with different morphologies, you could quickly assess organism behavior, investigate different food properties, and perform dose-response curves, and more. So yeah, just to end off, I hope I've been able to show you that the Reshape library helps you turn scattered experimental history into a reusable biological data library you can learn from. We're really excited about the progress we've shared today, and we're looking forward to even more impactful releases ahead. So with that, I will say thanks, and Mats, over to you if we have any questions.
Yes, I have a few questions prepared, but where you left off in terms of what we're working on next, I, of course, invited MA. So MA, could you fill the people in on what the Reshape product team is working on for the next foreseeable future?
Yeah, sure. I think Jen has already hinted on a lot of these things, but we've been really busy making sure we capture this very structured data, both on the experiment itself, but also on the metadata, as Jen has shown. And I think we've been exploring a lot about what we can then do with AI to actually extract insights from this data. And I think we're seeing with that also, the customers are trying to understand the value of bigger rollouts across the globe, across departments, to really build that data foundation that's needed to get insights. So we're busy expanding setups. But then from a software perspective, what we're really, I think the key thing for us will be giving these AI insights and bringing them back into the workflows in everyday labs and when planning experiments, when running experiments. And I think some of the key things are to help people get smarter about what experiments to run. So maybe we can help you point towards the highest probability experiment that gives the best results, or we can even present different parameters in your experiment based on our own data or on the Reshape library to get even better performance than what you've been planning so far. So really bringing all of those things that AI can give on a good data foundation back into the workflow of scientists.
Thank you. And maybe, as you can see on the screen, maybe a follow-up question June has is, if the AI learns when you run experiments, or is it locked down?
I could use a little bit of clarification, but the AI, it's kind of there all the time, right? So as you, with every experiment you run, new data is captured, and that is then fed into insights, and that will also fine tune what data, what experiment to run next, or maybe that will give insights into different parameters having different outcomes. So yeah, the AI insights are always there, and we'll learn together with you.
All right, thank you. We don't have any more questions, but just after this having been set live, Jen, from your experience back when you were in the lab, what would a tool like the Reshape library have helped you with, or what immediate benefit would you have gotten out of it?
That's a nice question. For me, I think the biggest impact is having the whole workflow in one platform fully contextualized. So in a normal lab setup, you spend so much time manually logging results, uploading files, data across experiments. But with the library, that friction sort of disappears, and you can move from sort of just maintaining data to actually making faster decisions, whether that be spotting trends early or narrowing down actually what is working. So hopefully it means less wasted time, fewer repeated experiments, and better use of lab resources.
All right, thank you. Shelley is asking if we'll share the slides. We'll share this whole recording afterwards. We just have to get it up on YouTube, but we'll share it later today or first thing in the morning tomorrow. Estimate time saved. I think that's a hard one. Probably depends on what you're running. Do you have anything?
Yeah, I think in actual like kind of human time saved, that's of course very dependent on what you're doing in the lab today. So we do look into this a lot with customers directly to get that clear, but that's a bit dependent on the case. I think the more interesting concrete metric in terms of time saved is probably time to result, and that is something we can very concretely drive down with those insights in the library. You can actually much better understand when you will get the results from your experiments. So I think in that sense, there's some very concrete metrics in time saved that are very interesting.
All right. I think, June, if you're fast, you can get the last question in, but otherwise, I think we'll round this up shortly. June isn't writing anymore. All right, guys, that was a quick sneak peek into the Reshape library. We hope you make the most of it, and then it can help you in your experiments out there. As said, we're going to be sending over the recording, and yeah, have a good one. MA, want to say goodbye to people? Any closing remarks?
Thanks for coming, and always never hesitate to get in touch with us to explore these topics in more depth.
Right. And Jen, thank you.
Thanks, everyone.
Thank you. Goodbye.
Bye.